Everyone, and thank you for attending the BC Delegation Spotlight today. I'm very much looking forward to hearing all of the presentations and want to thank you for joining us from around the world. As we begin, I'd like to acknowledge that I am joining this event from within the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. We would also like to acknowledge that others are joining us from the other traditional and unceded Indigenous territories from across this beautiful province, country, and around the world. Life Sciences BC is a not-for-profit society. Our members consist of academic and research institutions and local and global companies operating in the life sciences space, including digital health, med tech, med devices, diagnostics, therapeutics, and those companies that provide support and services to the broad life sciences ecosystem. We are funded through membership and the generous contributions of our sponsors and partners. Thank you to all of you for your support. We are very excited about today's event, the BC Delegation Spotlight, Bio San Diego. This event will showcase the amazing innovation taking place in BC, and we will hear from some of the companies who are attending the upcoming Bio San Diego conference from June 3rd to 6th. In attendance today, we have over 180 people, which is amazing, from Canada and across the world, including investors and government representation from the following countries: Canada, United States, India, Hong Kong, Thailand, Sweden, Lithuania, Japan, France, and Brazil. And we've also been told that many people will also be holding viewing parties to enjoy at a later time the great presentations we have today. So to start us off, I have the great pleasure of introducing Fazil Millar, Deputy Minister, Ministry of Jobs, Economic Development and Innovation. Previous to his current role, Fazil served as the Deputy Minister, Ministry of Energy, Mines and Low Carbon Innovation, Deputy Minister, Ministry of Jobs, Economic Development, Deputy Minister, Climate Leadership, and Assistant Deputy Minister with the Ministry of Advanced Education and the Ministry of Natural Gas Development. Before joining government in 2013, Fazil was Associate Editor of the Sun newspaper. Fazil holds a BA in Economics from Simon Fraser University, an MA in Public Administration from Carleton University, and has a marketing diploma from the Chartered Institute of Marketing in London, England. He also completed a certificate course on risk management at Harvard University and the ICD Rotman Director's Education Program with the Institute of Corporate Development. The floor is now yours, Deputy Minister.
Good morning, everybody, and thank you, Ryan, for the introduction. It's a great pleasure to be here to talk about British Columbia as we prepare for Bio International Conference in San Diego on June 3rd. Certainly, I do want to begin by thanking Life Sciences BC for organizing BC's delegation that will be attending Bio, and of course, hosting this Spotlight webinar. BC is certainly fortunate to have many of the world's most talented researchers and entrepreneurs working to solve some of the biggest health challenges that we do have. And at this point, I must admit, you take the risks in trying to create, do the research, trying to create products and help humanity. And the risk comes from the Italian phrase riscare, which means to dare. And I want to thank you for daring on our behalf in British Columbia. As you know, British Columbia has one of the fastest growing health science sectors in Canada. When you look at the GDP contribution, we saw a 30% gain when you look at between 2018 and 2021. We don't have the latest data yet, but certainly it's a growth story. BC Life Sciences companies, of course, have benefited from a rising tide of biopharma investment in the province. In the last 10 years, when you look at the BC biotech firms, there have been about 240 deals that have been completed with pharmaceutical companies with about total valuation of around $13 billion US. That's a testament to the innovative nature of our ecosystem. I mean, the province of British Columbia is committed to the life sciences sector. We, of course, will be attending Bio to promote BC's innovative ecosystem. Joining us at Bio will be at least 30 BC firms and organizations. And my goal in attending Bio is to meet with as many investors that I can to make sure that they understand that if you're hunting for molecules, you should be doing it in British Columbia because A, there are some great innovation, there are some great companies, and on top of that, there are much better valuations compared to Seattle, San Francisco, or Baltimore for that matter. So I know some of you are interested in what BC has to offer. So let me, as many of you know, we launched the BC Life Sciences Strategy, Biomanufacturing Strategy last year. There are many examples of the initiatives that we have put into place to support investment in people, in infrastructure, and, for example, funding over $331 million for the construction of a state-of-the-art 370,000 square foot clinical support and research facility as part of the new St. Paul's Hospital rebuild. On top of that, we have provided about $195 million to Michael Smith Health Research BC and Genome BC. We have also provided $20 million to Canada's Michael Smith Genome Sciences Center, all to support research and genome sequencing in collaboration with clinicians across British Columbia and the country. And with the federal government, we have made some other important announcements, as many of you are aware, to build out a new national biomanufacturing training center at BCIT, which will train up to 700 people every year to make sure that people have the skilled talent they need to thrive in British Columbia. And of course, we supported AbCellera Biologics for their $700 million expansion for the state-of-the-art biotechnology campus in Vancouver. The government of Canada is also making significant investments to grow BC's life sciences ecosystem. In early May of this year, Canada announced an investment of $140 million in projects associated with Canada's immunoengineering and biomanufacturing hub. It's a national research hub led by the University of British Columbia with more than 50 academic, industry, and not-for-profit healthcare sector partners. We will, of course, support that endeavor as well in British Columbia. Earlier this year, we also announced $32 million to support an expanded capacity for cancer research at Triumph, which is also situated at UBC. It's the Canada's National Particle Accelerator at the University of British Columbia. We have certainly, on top of that, we have invested in the expansion of clinical trial capacity, particularly for stage 1 clinical trials with Providence Health, along with the endowed chair at UBC to oversee the clinical trials. Our government is also, of course, government of British Columbia is also investing about $12.5 million to increase the number of wet lab space that we have so that firms that are getting out of the university setting have a place to go in terms of clinical trials, or in terms of making sure that their work can continue. We also have set up a $500 million strategic investment fund, which has now started making direct investments into innovative BC companies like Clarius Mobile Health to help them expand their global outreach. So I think, let me close by saying that we are extremely proud to be able to support our companies at BIO in San Diego on June 3rd and raising the profile of British Columbia. And I do want to thank Ryan and Wendy Herbert at Life Sciences BC for being true champions of the sector. And I look forward to seeing you all at BIO and hoping for success in terms of attracting the kind of investment dollars that you would need in order to succeed and continue your research projects. So thank you very much, Ryan.
Thank you very much, Deputy Minister, for your very kind remarks, and we really appreciate your continued strong support of the life sciences ecosystem, and we'll look forward to seeing you soon in San Diego. So I'd now like to present an overview of life sciences in BC, starting with a snapshot of the province of British Columbia, as we have many international guests joining us today. BC is Canada's 3rd largest province and is located on Canada's west coast. It is part of the Cascadia Innovation Corridor, including Washington and Oregon. As of January 1st, 2024, BC has a population of over 5.6 million, with a growth rate of 3.3% over last year. This was the highest growth rate the province has seen since 1971, and you can see why with our stunning land and cityscapes. We have a strong GDP at $304 billion as of 2023. BC is also home to some of Canada's top universities and research institutions, including the University of British Columbia, where 20% of all Canadian spin-outs originate. The BC life sciences sector is headlining national news. We were recently featured in a Globe and Mail article by Sean Silcock, profiling some of the unique regional stories and companies that are making it big here in BC. Brian Bloom, co-founder of Bloomberg & Company and the firm's chairman and CEO, was quoted in the article noting how Vancouver in particular stands out. We are on track to become a globally competitive sector, recognized for our innovation and uniquely collaborative culture. We have the fastest growing life sciences sector in Canada. Biotechnology alone has attracted over more than 13 billion US investments in the past decade, and many more deals to come. There are 28,500 plus employed in the sector and about 2,000 companies. Since 2019, the life sciences sector has achieved 27% growth in GDP, contributing 3.1 billion in 2022. And life sciences currently outpaces the growth of BC's overall economy, something we can all be very proud of. Here are examples of some companies mentioned in the Globe and Mail article and others that are built in BC success stories. We are home to Canada's largest biotechnology company and employer, Stem Cell Technologies, and Canada's largest medical device technology company, Starfish Medical. We have several BC-based companies with over $1 billion in market capitalization, including Accelera, and with over $2 billion market cap, including Xenon Pharmaceuticals. Some of the biggest deals that are happening in BC, including Aspect Biosystems' $2.6 billion collaboration with Novo Nordisk, and Chinook's $3.2 billion acquisition by Novartis, both huge success stories that we can all be very proud of here. We're a world-class biopharmaceutical cluster. Some of our strengths include antibodies, regenerative medicine, nanomedicine, or lipid nanoparticles, genomics-based precision medicine, medical technology, and digital. Many companies are integrating machine learning and AI into their workflows and as part of their innovations. A large number of BC companies in BC are also working in the oncology space. Last year, there was a pioneering co-investment, as the Deputy Minister mentioned, of $700 million by the Canadian federal government, British Columbia's government, and Accelera to expand Accelera's antibody drug development capabilities and infrastructure here. And I was recently there for a meeting, and it's a fantastic facility, and look forward to seeing the future developments there. Aspect Biosystems 3D bioprinting technology could be transformative for patients with type 1 diabetes and liver disease. Acuitas, who we'll hear from later today, developed a lipid nanoparticle technology, which has enabled the treatment of a rare genetic and otherwise fatal disorder. Their technology was also used in the Pfizer-BioNTech COVID-19 vaccine, which has protected billions of people in more than 180 countries. The innovation was based on the decades-long work of Dr. Peter Cullis. These are just a few examples of the high-caliber life sciences innovation happening here in British Columbia. And you will hear from 7 other BC companies and organizations that will be joining us in San Diego. So I'm very excited to announce and to introduce our first speaker. I would now like to welcome our first presentation from Dr. Emily Chen, Associate Director of Business Development at Zymeworks. Zymeworks is a global biotechnology company committed to the discovery, development, and commercialization of novel multifunctional biotherapeutics. Zymeworks' complementary therapeutic platforms and fully integrated drug development engine provide the flexibility and compatibility to precisely engineer and develop highly differentiated antibody-based therapeutic candidates. I now welcome Emily to begin your presentation.
Thank you, Ryan. Just to check if everyone can see my screen okay? Is my presentation up? Okay,
Yes, that's great.
Okay, thank you. So my name is Emily Chen. I am honored to be here representing Zymeworks to give you a corporate overview. So Zymeworks is dedicated to transform the standard of care of difficult to treat diseases. To achieve this goal, we're heavily leaning on our fully integrated R&D engine, which is shown in the middle of this slide. And our R&D engine is composed of 2 fundamental platforms. Starting from the bottom half of the circle, we have multiple toolkits to support our protein engineering capabilities. And using all the toolkits, we are able to design and develop novel differentiated multi-specifics. The top half of the circle represent our medicinal chemistry capabilities to design a novel linker payload for ADC use. Combine our medicinal chemistry and protein engineering capabilities, we believe we have a holistic view into ADC design, and which can help us get to truly meaningful and differentiated ADC therapeutics. We set up a goal to, or a 5x5 strategy, which is our goal to deliver 5 INDs in 5 years. And this slide gives you a quick update on the current status of our 5x5 strategy. We have 2 INDs planned this year, and one coming up soon is 0171, is a T-cell engager targeting mesothelin. And the second IND this year we're planning to do is our first topoisomerase ADC targeting folate receptor alpha. We have 2 more INDs coming up next year. Both utilize our topoisomerase payload as the payload, and one targeting NaPi2b, the second one targeting GPC3. So now let me try my best to give you a little bit teaser into our platforms, starting from our ADC side. So again, I think our Zymeworks is sitting in a unique and differentiated position as we have a holistic capability to look both into the ADC from the linker payload side and also the antibody side. And we believe that only to optimize and integrate all key aspects of ADC will enable us to design a valuable and successful ADC asset. So starting from the antibody side, you need to optimize your antibody for ADC function when you're thinking about your selection here, particularly to have optimal internalization, tumor penetration, and payload delivery. It's not just about antibody affinity. Similarly, on the linker payload side, it's not just about to push a super high state stability of linker or a super strong potency on the payload. You need to find a sweet spot of both to achieve the balance of efficacy and safety. To make it even more interesting or complicated, each indication and target, you need to think strategically about how to combine all 3 pieces together. So this is not unfortunately, a one-size-fits-all type of solution here. And this table highlighting the 3 near-term ADC assets we're developing right now, and I will use 0191, which is our leading topo ADC, as an example to showcase how we apply our philosophy into development of a novel ADC molecule. This is the target of this ADC is folate receptor alpha, and it has a DAR8 with topoisomerase as the payload. And I think at the first sight, we believe it will have a best-in-class potential as we see a great 60 mg per kg non-human primate MTD. Just to showcase a couple key design considerations around 0191, and the internalization, when we compare 191 with multiple clinical or approved folate receptor alpha ADCs, across the board, we see better internalization, better payload delivery, and tumor penetration. And all of this translate into greater anti-tumor activities, not only in ovarian cancer across multiple folate receptor expression levels, but also other tumor types such as non-small cell, endometrial, and TNBC. And we highlighted this data in a poster at AACR this year, so I will definitely encourage you to check it out. Moving on to our multi-specific platform, and more specifically, our strategy around a tri-TCE co-stimulatory therapeutic program. I think, as we all know, the first generation T-cell engager focusing on activating the signal 1 or activating T cells through signal 1 through CD3, but this is not, I guess, the optimal combination to get the full activity out of T cell, and you need CD28 to combine with signal 1, have the optimal activity. But this is tricky, right? Because you need to balance the dial. If you dial it too far, likely you will see a strong toxicity profile through CRS or immune-related adverse event. So what we are trying to accomplish at Zymeworks is that to do a thorough job to find the optimal combination of the right geometry with the right CD3 and CD20 affinity. So what we did here is we generated a large library taking advantage of our asymmetric platform, a large library of tri-TCE formats, and also testing a bunch of CD3 and CD28 binders with different affinities. And hopefully through this exercise, we will be able to find the optimal combination of the three to get the desired profile of a tri-T-cell engager. And this slide showcases our proof of concept on data. And we developed a tri-TC co-stim targeting Claudin 18.2, which is highlighted in blue across all 4 panels. I don't expect you or myself to go into all the detail of the data during today's presentation, but the key takeaway point of this slide is that across the board, when we compare our tri-TC co-stim molecule with the traditional bispecific TCE without CD28 or the clinical benchmark from Amgen, across the board, we see better cytotoxicity, cytokine release, anti-tumor activity, T-cell expansion, T-cell proliferation, and also last but not least, a better CRS profile. So using this proof of concept data, we're actively pushing our 5th 5x5 assets and as a tri-TCE candidate. So beyond our 5x5 strategy, we also have an advanced portfolio strategy, which really means that we're looking further beyond the things that we do right now and try to explore more innovative, more advanced technology to find novel targets, novel payload, novel technologies for ADC and multi-specific therapeutics. And beyond oncology, we're also thinking to expand our capabilities into autoimmune and inflammatory diseases. And the last piece here is about IO strategy. We're actively also seeking some IO assets that can be complementary with our existing pipeline activities. Just to wrap up my presentation here, to give you our partnering strategy. And as you see, we have a lot going on at Zymeworks, and we are open to have partnering conversations across different aspects of our platforms. And first of all, we are definitely excited to work with partners strategically to develop molecules together, not only on TCE, but also maybe on ADC side. And on our advanced portfolio side, we are also very excited to explore jointly discover some novel bispecific ADCs and absolutely in any other aspect of it, our advanced portfolio is open for conversation as well. For our 5x5 strategy, 5x5 portfolio, we're also seeking capable partners to collaborate on the development. There's always room and opportunity to accelerate the clinical plan, to expand the patient population, or maybe find interesting indication to target different regions. So yeah, I will end my presentation here, and I welcome any questions from you guys, and looking forward to meet you all at Bio in person. Thank you.
Great. Thank you very much, Emily, for that. It was a great presentation, and also wanted to thank Zymeworks, as they will be hosting our sold-out summer social next month. So we're very much looking forward to that. Now, our next speaker is Dr. Anthony Fejes, co-founder and CEO of H2O Biosciences. H2O Biosciences is a deep tech company that works at the interface of physics, chemistry, and computer programming to develop advanced computational drug discovery methods. The core technology uses a unique physics-based model to accurately represent the behaviors of atoms and molecules that better mimics the natural world and does not rely on training data. H2O's goal is to leverage the core software Atom Forge to transform drug design and expedite drug discovery, particularly for challenging to target proteins. The floor is yours, Anthony.
Thank you, Ryan. Thanks for giving me the opportunity to talk among such an awesome group of companies presenting today. My goal today is to give you guys an opportunity to see what we're working on and to show why we're a lot different than all the other companies in our space right now. Oops, I'm sorry, get rid of that. So just a quick overview of H2O Biosciences. Of course, I'm Anthony Fejes, the CEO and co-founder. We started the company in October 2020 and have raised about $1.5 million US to date. We're just currently in the process of closing an extension to that as well. The funds have allowed us to grow our team to 9 people and are looking to grow that aggressively in the near future as well. We're currently preparing for commercialization, and we've had some great successes with our technology in the last couple of months, a few breakthroughs, and that's pushing us in the right direction, which I'll tell you a bit about in a moment. And we're also very proud to be members of the San Francisco J Labs. That's part of the J&J Innovation Arm, and that was since January of this year. So the goal of our company is fairly clear for us. We want to be able to do what no one else has done before, which is be able to do high accuracy computational drug discovery or drug optimization. So currently, if you're using any of the platforms on the market, you would have a difficult time making a single nucleotide switch or switching a small side chain on a drug and asking the question, what is the outcome of making that change? None of the competitors on the market right now can do that with high accuracy, and we believe there's an opportunity for us to do so. Ultimately, the reason why we want these high accuracy energy predictions is because we want to be able to move in a direction that others have not been able to move so far. There's a huge amount of targets currently that no one is able to drug. Generally, we just classify them all as undruggable proteins or undruggable targets. They generally lack clear binding sites. They have or shallow binding sites, or there are large surfaces to work on. They can be fairly complicated chemistry to work on. And without the high accuracy platform, it can be very difficult to develop any drug that will attack or inhibit those molecules. So we believe that our platform, Atom Forge, which I'll tell you about in a second, will allow us to work on these novel medicines for difficult undruggable targets. And ultimately, this is something that's really important in the age of personal genomics, or where we have the opportunity to work on generative AI designed targets or structured targets that we're interested in. So our core technology comes in 4 components. The first is the core physics. We spent the first 2.5 years of the company building our core physics module. That allows us to accurately represent the physics of what's going on at the core level. Around that, we wrap what's called a parameterization, and that's where the physics model accurately maps to the chemistry. If you have a good parameterized physics model, it allows you to make predictions about what's going on and how all molecules are structured. The closer this gets to completion, the closer you get to having a platform that allows you to make accurate predictions about structures and interactions. Around that, we wrap what's called molecular dynamics that allows you to take what is a snapshot of an interaction and convert it into essentially a movie. So make predictions about how things interact over a period of time. The final aspect that we're working on currently, which we expect to have for the end of this year, is what's called a free energy perturbation or free energy calculation algorithm. This is something that's been very popular in the last 5 years or so, and we believe that we have an algorithm that's going to allow us to make higher accuracy and more efficient predictions about how drugs interact with their targets. And that's the focus of what we've been working on. The ecosystem into which we're working has 2 major components of it. One is the scalable side, and the other is the accuracy side. Typically, a lot of the competitive products out there right now come from the molecular modeling world. They're usually very scalable. You can work on biology-sized projects on them, but they don't typically have the high level of accuracy required to make single nucleotide changes for a target, so understanding how an oncology mutation might affect the target, or to be able to make small modifications to drugs. On the other side of that platform, you also have the accuracy. That's quantum mechanics based. Generally, they're extremely accurate, but they lack the ability to scale to biology-sized problems that we're interested in. We also don't consider ourselves directly competitive with generative AI. We consider them more of a complementary technology. We're working either upstream or downstream of them. We can generate the data used to train AIs, or alternately, we could validate the data being used generated from those AI platforms. What's really interesting about our platform is that we've gone the opposite direction of most of our competitors. We've really focused on getting back to the chemistry that's underneath. So in nature, there's really only 3 types of carbons. We talk about hybridization states, so sp1, sp2, sp3. We've gone back to that type of chemistry, where our competitors typically are unable to get that level of mapping with direct chemistry. They'll use many different types of atoms. So they'll use 3 or 4 different types of carbons to represent the aromatic carbons or in a long chain. We don't need to do that. All of our carbons act the same regardless of their environment, and that's a unique feature we have. We're very, very proud of this fact. It allows us to get much closer to the original physics and chemistry that lets us do less parameterization and clearer design work at the end. We can show this at the aggregate level. OpenFF is one of our bigger competitors. This past year, they released a nice paper where they stacked themselves up against what was the state of the art previously, GAF. In 2002, you can see they had a success rate of about 18% of their molecules have no error. And there's some broad shoulders out there on the side. I'll talk a bit more about that in a second. In 2023, OpenFF showed that they had about a 2 or 3% improvement to that core piece, which is a significant improvement. However, we can also look at our technology as we've recently recalculated this benchmark, and we can show that we're now about 24, 25%. So what's really important is, of course, that this shows that we have fewer error in the molecules that we calculate. So we have more molecules with less error. But it's also interesting that we're able to have less molecules with more error, which is equally important. This is a statement to how accurate our platform is going. So what this actually looks like at the largest state is referring to another figure from the OpenFF paper. They showed a molecule that they cannot calculate, or they calculate poorly. In our case, we can demonstrate that plugging the same molecules in where OpenFF is failing allows us to generate the right structures at the end. This is important, for instance, if you're working on a drug design problem. If you're interested in understanding or screening your molecules and looking for the ones that fit best, making sure that you have the right structures is a critical component of that. And of course, calculating the interaction energies at the end. If you fail in this respect, you end up with a lot of false positives or false negatives, and we can help reduce that going through that process. Ultimately, our platform is being used for two different purposes, or two different business models. The first is that we are looking currently for strategic partnerships and places where we can apply our platform to help other companies with their structured drug design projects. We are not currently looking to design our own targets, but in the long term, we anticipate that having a platform that's fully validated will likely go in that direction, where we can design our own molecules and own the IP ourselves going forward. Right now, the future of our platform is fairly clear. We're currently working on protein ligand binding. We're generating clear data that shows that we're heading in the right direction. We anticipate having our full FEP tool, FEP type tool ready by the end of this year. That will allow us to work on drug design problems directly. And we're looking currently for partners who are interested in working on that. And in the long run, we expect that this will take us in the direction of undruggable targets. So we're very interested in looking towards partnerships and getting feedback from potential partners. If you're interested in seeing if our technology could work for your platform, please let us know. And if you're interested in talking about drug design or drug optimization with us in the long run, please feel free to reach out, and we look forward to seeing you at Bio. Thank you.
Thank you very much for your presentation, Anthony. And again, congratulations on being part of JLab San Francisco. When we were down there for the JP Morgan Week conference, we had the chance to tour that facility, and it's an amazing facility. And also during the upcoming Bio San Diego, we'll be leading a delegation to visit the JLab's San Diego facility, which was the original one just north of San Diego in La Jolla. So next, I'd like to welcome Suzanne Gill, President and CEO of Genome BC. Genome BC is a not-for-profit organization supporting world-class genomics research and innovation to grow a globally competitive life sciences sector and deliver sustainable benefits for British Columbia, Canada, and beyond. The organization's initiatives are improving the lives of people throughout BC by advancing healthcare in addition to addressing environmental and natural resources challenges, while fostering understanding and appreciation of the life sciences among educators, students, and the public. Thank you for joining us today, Suzanne. Over to you.
Great. Thank you, Ryan. I think folks can see my screen.
It's just not quite in full screen.
Great. Thank you. There we go. Good.
Perfect. That's
All
great.
right.
Thanks.
Thank you, Ryan. So really, I'll just spend this time to talk a little bit about what we do at Genome British Columbia. And as you heard from the Deputy Minister at the beginning with his remarks, we're very grateful to have the support of the province. And we really are one of the organizations among a few that have created the foundation for BC's life sciences sector here in BC over a couple of decades. So I'll talk a little bit more about what we do and who we are and how we go about our work. We are an organization that invests in genomics research and its applications in 4 main areas. And I'll just go to the next slide. There we go. Obviously, one of the biggest investment areas for us is in human health, and that is really around the prevention, diagnosis, and treatment of disease using genomics at its core and access to the best-in-class technology to do that. Second big area of investment is around food security, and that does mean we are looking at how do you create drought and disease-resistant crops so that we have, food in the future given a change in climate. We also have quite a significant and growing investment around the environment, which includes sort of sustainable resource development and also benchmarking the health of our environment, understanding our species at risk and how to mitigate and manage the changes that are going on there, given industrial activities as well as climate change. And then our fourth big bucket, I would say, which does cross into the other areas as well, is really around how do we support climate adaptation for our communities in our regions that include the natural resources sector. So how do we ensure we are planting and developing trees that have climate adaptive traits, given it's going to be several degrees warmer over time, and we have a healthy and productive forest, and all the way downstream to bioproducts and other things like that. Now, really, who are we? So our mission really is to apply the power of genomics to pressing societal and economic challenges facing our industries. And really, one of the big things we do there is by convening the best people in our post-secondary institutions, our research institutions, with government and with industry that really have some challenges facing their growth. And we do it by doing four big things. We invest heavily, and I'll give you some examples in a minute, in world-class research. We provide the best access to the technologies through our platforms, and we have a number of them in BC, including one that was mentioned earlier, the Michael Smith Genome Sciences Center. And we also engage the public and key stakeholders in discussions around the responsible application and use of genomics as the field evolves and as these applications get put into practice in the clinic. And we have a suite of activities that we undertake to accelerate the adoption and reduce the barriers to deploying genomics into practice. And that does mean we get into the space around education, inspiring our future generation of scientists. It means that we are also into professional education. How do you accelerate, as software applications include genomics tools, and how do we ensure that the users in the clinic are able to use that and interpret that in the right way for the benefit of patients. Now, this is just a slide to give you an idea of the scale of our operations and our portfolio over the last 23 years. So it's been a long time. We have almost 550 projects across those different sectors, as I mentioned, and over $1 billion of investment to date. And I'll talk a little bit more how we've connected the dots really now to what that great article in the Globe and Mail alluded to BC's biotech scene. So there's more of this on our website if you'd like to take a look. I do want to say that Genome BC, its origin story really, as some of you may know, was Canada was not part of the international sequencing effort to sequence the human genome at the cost of $3 billion many, many decades ago. But where we are today, because of government investment, is we are part of a national network of genome centers. We're all independent, private, not-for-profit entities. And BC is probably one of the larger ones, in part because of the partnership we have with the province and other industry partners here. But that does mean we coordinate across the country around areas of strengths and bring and draw in, if we need to, from other regions to fill that gap. And just to highlight, this is how we approach our work. And as I mentioned, we do a lot in research. We call it research and innovation because I'll talk a little bit about our efforts in commercialization and entrepreneurship as things are maturing in genomics and in life sciences industry in BC. We have a vehicle now to support companies directly, and I'm sure someone will drop something in the chat about where that is. But also, as the field evolves and genomics is being put into practice, we are making more investments in the work around engaging the public, education and outreach, discussing with the right policy analysts, what does it mean if you deploy a new technology into practice, whether that's human health or in the environment. So that interface really between genomics and society is becoming ever more important for Genome BC. I mentioned a little bit about partnerships. So this is just a small example of how we work. For every dollar we receive from the province, we leverage it and attract at least $3 from others. A part of it is from the federal government, but most of it is from the private or public adopters or technology product innovators that will take on using genomics or come through our programs. So today, which is a great story, is Genome BC, after 23 years, can connect the dots from investing in research and technology development to 5 of Canada's top 10 biotech firms, and they were mentioned earlier in this. Someone else mentioned it earlier in the presentation, but that is the likes of Abcelera, Xenon, which was one of our early investments, Aspect Biosystems, etc. So very proud to be part of that growth, and we look forward really to the next wave of what we do. So most of our funding is deployed through our programs, and there's lots of programs that we run. I think we're running about 6 or 7 right now that are active, and you can find the information on our website. But we do work with others in the ecosystem. Most of our funds go to the universities, the research-intensive universities. We are seeing a growing partnership with some of the smaller ones, so the likes of KPU in agriculture, or Thompson Rivers University in Kamloops around mining. So the smaller ones tend to have stronger links with industry. And as we look to solving their challenges with genomic solutions, we see those smaller ones coming through our application-oriented programs. So a great opportunity there. But mostly in human health, we deal with the Faculty of Medicine at UBC and the health authorities and the hospitals directly. Just some of the impact Genome BC has had after 23 years. And in the early days, we used to describe the opportunity for life sciences in BC was that we would be as big as the IT sector at some point. And I could say proudly that we're on track to do that. We have over 51,000 jobs just from the analysis that we've done at Genome BC for our impact. So that's much bigger if you take in others, and contributed almost $5 billion to the province's GDP. What's interesting is we are at a point of inflection. The impact of our activities in the last 5 years through our investments through our programs and our companies has meant that the impact in the last 5 years is greater than the sum of the previous 15. And so now is the time to, in my opinion, to continue building on that investment in supporting the companies that are coming out of the life sciences sector. And there's programs for that the feds are running and the province is running. So very, very good to see that investment. And of course, LSBC's role in that in the life sciences strategy. These are some of the companies advanced by Genome BC. I think I mentioned some of them earlier, but it does take a long time to see the fruition of research coming out of the universities, but super fantastic to see some of the names that you'll recognize here. And some of them are on this panel today. So I'll leave that with you. Lots of stories there. And I would encourage, there was the link I put in the chat around the biotech scene in BC and a little bit of its origin story. And it's a great story. So just really quickly before I wrap up, this is how we go about our work. So Genome BC has an industry innovation fund to support sort of the commercialization and entrepreneurship side of the business. We have a $20 million fund. It's been active for about 8 years now. We have made investments in 14 ventures to the tune of about $16 million. It is a debt instrument explicitly right now, no equity. And you can find more information on our website for that. And happy to answer any questions, but I think I probably wouldn't be the one to talk about all the details. I'd refer you to Pat Brady, who's on our team leading that fund. We also partner with some of the accelerators, incubators, and supports for entrepreneurship that are in the ecosystem, and some of them are here. So our most recent one is the Creative Destruction Lab, but you'll be familiar with probably entrepreneurship at UBC, which was really one of the founding investments we made in supporting entrepreneurs through that program. As I mentioned, one of our proudest programs that we have in terms of societal engagement, lots of stats on this slide, but what I would like you to take away from this is that we engage the students across this province in all corners of our province through our Gene School program, largely through the support of the science teachers that are in our schools for grades 9 to 12. So fantastic resource there available to anybody who is interested. And you can go find more on our website. I'll just leave you with a few highlights of the things we've funded recently in cancer, which really leads our investment in the area of human health. We've really done a recent investment in hereditary cancer, which is really around trying to prove out a concept of a new technology to be deployed in the BC Cancer Agency. And very proud it was a multi-million-dollar project that includes obviously the Genome Sciences Center that does a lot of the sequencing is integrated through the Cancer Center. We also were heavily involved, nothing like a pandemic, to provide an opportunity to accelerate the adoption and deployment of tools related to surveillance around new virus variants that may be of threat to public health. So we just announced actually a new project with the BC CDC and the Public Health Lab to keep that surveillance network alive and to use the latest tools for the public health officer to be able to do that. And in rare disease, our Rapidomics project, another $5 or $6 million project we just announced a few quarters ago, also using the Michael Smith Genome Sciences Center, and is based at the Children and Women's Hospital, really to identify genetic disorders in infants at a young age. And we also, in parallel, are running our Silent Genomes project with funding from Genome Canada, which looks at Indigenous communities and Indigenous populations that are really not able to access the same level of care. We know health inequity is huge. So we are trying to move the dial on changing that through the Silent Genomes project. And just because I'm a big fan of making sure you take away something other than the things we do in human health, we do have some work going on. It's been going on since 2015 with the BC Centre for Disease Control, Ministry of Agriculture, Canadian Food and Inspection Agency, and industry around how do you develop a surveillance system for avian flu that may become a threat to public health. So we're actively working with those stakeholders on that. Climate change, wildfires, huge area of interest for us. So we are working with the Ministry of Forests and other stakeholders around how do you have climate adaptive programs to develop new seedlings really to plant to replace many of the trees that we're losing through wildfires and forest practices right now. And then finally, a very interesting, really cool project I'll tell you about. We are really taking the learnings and expertise we have built up over 2 decades working in human health to be able to really apply it to some of the significant challenges in other industries. Right now, critical mineral strategy is out in Canada. We'll have one soon for BC. So what does it mean that where can genomics play a role in terms of providing solutions to those industry challenges? One of the projects is the Mining Microbiome Analytical Platform, big mouthful, but it is looking at taking 15,000 samples provided through our partners, that's Teck, the mining giant, as well as Rio Tinto, to identify what microbial communities are in the environment that we could really look at, sort of like a drug development pipeline for environmental remediation. Where can we have clues from these microbial communities and what activities are happening there that provide us with some indication of where to look for solutions that we could develop over time for remediation strategies or mine reclamation solutions. I think that's my last slide. So thank you so much to LSPC for letting me join you today. And I hope you enjoy the rest of the presentation.
Great. Thank you very much, Suzanne. That was a very fascinating presentation, lots of great stats. And just also want to thank you, Suzanne, for all your amazing work as a board member of Life Sciences BC. It's very much appreciated. And just also a reminder that this webinar is being recorded, and so we will be sending out the link tomorrow. And we also encourage you, if you have any of your colleagues or other, either investors or government contacts that you feel would be interested in this presentation, please feel free to forward on to them as well. Appreciate it. Next, I'd like to introduce Dr. Hal Gunn, the CEO of Q Biologics. Q Biologics is a clinical stage company developing a novel class of innate immunomodulators designed to transform the treatment of chronic inflammatory diseases, metabolic disease, and cancer, the optimization of health and the extension of health longevity by restoring all the cells, pathways, and roles of innate immunity in a targeted disease order. To use site-specific immune modulators, SSIS, aren't designed to simply treat disease. They are designed to restore the full depth and breadth of normal immune function in a targeted diseased organ to transform the way we think about the treatment and prevention of disease and optimization. Take it away, Hal.
Thank you, Ryan, and Life Sciences BC for the invitation to speak today. Much appreciated. Ryan, if I'm not sharing my screen appropriately, please let me know. But my goal of this presentation is to communicate the very novel nature of what we're doing at Q, the transformative potential of our platform, and the potential to change the way we think about the treatment of disease and the optimization of health. Our platform is health restorative. It isn't designed to simply treat disease. It's just designed to restore the full depth and breadth of immune function in a targeted diseased organ to restore health. And it does that by training the innate immune system. And I'll talk more about what that means and how that's achieved and how very different that is in the way we usually think about the treatment of disease. Our platform's designed to be safe and targeted and targeted to the organ of pathology. So first I'll describe the roles of innate immunity. Many of you already know those roles, but just to give a quick overview, and then to describe what the problem is that our platform uniquely solves. Innate immunity plays a very important role in the body. It plays an essential role in controlling inflammation, regulating metabolism, maintaining barrier function and a healthy microbiome, mediating autophagy, modulating adaptive immunity. If the innate immune system isn't working properly, the adaptive immune system can't work properly, and clearing infection and cancer. So this is a very wide range of important roles. The problem is that chronic inflammation and cancer and metabolic disease all dysregulate innate immunity. And then a vicious cycle is set up where that dysregulation of innate immunity and all of its roles worsens chronic inflammation that then worsens the innate immune dysregulation. And you can't address that issue safely and effectively with monoclonal antibodies or small molecule drugs. You need to do it by training the innate immune system, and that's what our platform does. By analogy to muscles, muscles lose strength and resilience if they're not properly exercised. The same is true with our innate immune system, and our innate immune system is trained through beneficial exposure to bacteria. If the innate immune system doesn't get that exposure and training, it becomes dysregulated. And as you can see in this graph, over the last 80 years, as the incidence of acute infection has dropped dramatically because of vaccination and improved hygiene, which is a really great thing because acute infection used to be the leading cause of death. One of the unintended consequences of that is a rise in immune-related disease because the innate immune system is no longer getting the training that it needs to maintain its resilience and strength. We've discovered the underlying mechanism of that organ-targeted training. The innate immune system is trained in an organ-specific way. And how does that translate to a treatment? And what is our platform? So our platform is derived from inactivated bacteria. You can activate this mechanism with live bacteria, but of course that's a risk. And so we have preserved the components of the bacteria that create that response, but they're made from inactivated bacteria, so they can't cause infection. But what they do is they target that innate immune training in a specific organ or tissue. And the mechanism of that is through hematopoiesis, trained innate immunity, and organ-specific chemokine release. What that allows us to do is to target newly produced, newly activated, trained innate immune cells to a targeted diseased organ to replace the dysfunctional innate immune cells that are there in disease and restore all the roles and functions of innate immunity that underlie those diseases. What we call site-specific immunomodulators, or SSIs, are administered by are self-administered by patients themselves by simple subcutaneous injection. There are 6 steps to the mechanism. The SSI is self-administered subcutaneously. That there are then components of bacteria where they shouldn't be. That causes the release of cytokines that activates myelopoiesis in the bone marrow, with release of newly activated trained innate immune cells that then are targeted to the organ or tissue where the bacteria from which the SSI is derived is endogenous. Essentially, the SSIs are carried systemically, and it's only in the targeted organ that has seen that bacteria many times before that trained innate immune cells release chemokines and then draw those newly released trained innate immune cells into the targeted organ to clear pathology in the targeted organ. It's a very unique mechanism, and what that allows us to do is restore and train innate immunity and return to normal function all the roles of innate immunity in that targeted organ. Here's an illustration of its effects clinically. We've completed 2 small phase 2 studies in Crohn's disease. People with Crohn's disease have a dysregulated innate immune function. The best way to measure that is with IL-1 beta, which is the classic cytokine to measure innate immune training. You can see at baseline, the people in this study had lower innate immune training, meaning innate immune dysregulation. And after 16 weeks of treatment, you can see substantial improvement in innate immune function and the ability of the innate immune system to play all of its roles, including in the context of Crohn's disease, barrier function, bacterial clearance, autophagy, and clearance of dysbiosis and regulation of the microbiome. That then is correlated with the clinical response we saw in our studies, clinical symptom improvement, endoscopic response, and histological response. This is a very different way of treating Crohn's disease. Currently, Crohn's disease is treated by treating the end result of the disease, which is the overreactive adaptive immune response. That overreactive response is to bacterial invasion, which is a result of a dysregulated innate immune system. We're very excited about the application of our platform in IBD as a very different way of treating both Crohn's disease and ulcerative colitis. But our application is much more broad than that. This is an example. This is in the treatment of metabolic disease. I'll tell you a quick aside. We did a 6-month toxicology study. We found no observed adverse effect levels in all of the 3 doses that we treated. But interestingly, the mice had improved glucose control. And I joke with my team, our treatment has side effects, but they're positive side effects because we're restoring all of the roles of innate immunity. So it's a very different way of thinking about what side effects might mean when you're thinking about small molecule drugs or monoclonal antibodies that have off-target negative effects. Our treatments are designed to restore health. And here we did a collaborative study at McMaster University with the Canadian Chair of Metabolic Disease. And here you can see improved glucose tolerance tests. We also demonstrated improved insulin sensitivity, and importantly, improvement in body composition with a decrease in fat mass without a loss in lean body mass. Innate immune dysregulation underlies many important disease categories: cancer, chronic inflammatory disease, metabolic disease, chronic infection. These are all very large markets, and our platform is uniquely positioned to create drugs that are immunomodulators that address all four of these important areas. And also importantly, Q is positioned to lead the emerging markets of health optimization and extension of healthy longevity and prevention. So the unique nature of our platform that is targeting the restoration of innate immunity and the impact of innate immunity on overall health. Here's our clinical development program across a wide range of important diseases. We've completed 4 small phase 2 studies with proof of principle in Crohn's disease and ulcerative colitis, and immunological proof of principle in lung cancer. We have 2 randomized placebo-controlled trials underway. Importantly, there are indications for which there's no current treatments because our platform is uniquely positioned to have benefit in those areas. One is in perioperative colon cancer. Surgery suppresses and dysregulates innate immunity, and we've shown in animal studies that our platform prevents that from occurring and improves survival in perioperative cancer surgery. This study is in late stage colon cancer in patients undergoing resection of liver metastases. It's currently enrolling, and we expect milestone data at the end of 2025. Our second current randomized placebo-controlled trial is in immunosenescence, which is the decline in immune function with aging. Decline in innate immunity with aging predisposes to the development of cancer, infection, and chronic inflammatory disease. COVID-19 was a very good example of that predisposition, where it was the elderly that were at greatest risk. We're doing a study to see if we can restore innate immune function in the elderly, which has really important implications for extension of healthy longevity in the elderly and the prevention of diseases and the progression of diseases in the elderly. We're raising a minimum of 25 million. The milestones would be the completion of the 2 studies that I just mentioned, initiating a phase 2 randomized placebo-controlled trial in inoperable pancreatic cancer, enabling phase 3 manufacturing process development, expanding our team, and extending our runway through to 2027. The company has been de-risked in a number of important ways. As I mentioned, we have clinical proof of concept. We have a very good safety profile in the more than 360 patients that we've treated. The manufacturing processes are easily scalable and validatable, and we have additional proof of principle across a wide range of diseases preclinically. We've raised $39 million US to date, and we're seeking a minimum $25 million Series B funding round. We have 68 granted patents across 7 patent families, and we're expecting the milestone data from our immunosenescence study at the end of this year and our perioperative study at the end of next year. So thank you for listening attentively, and I'll look forward to seeing you all at BIO.
Great. Thank you very much for that very interesting presentation, Hal. Next, we have a presentation from Dr. James Halliwell, co-founder and CEO of Eupraxia Pharmaceuticals. Eupraxia Pharmaceuticals is a clinical stage biotechnology company focusing on the development of locally delivered extended release products that have the potential to address therapeutic areas with high unmet medical need. Diffusifere, a proprietary polymer-based microsphere technology, facilitates targeted drug delivery with extended duration of effect and offers multiple highly tunable PK profiles while being compatible with multiple active pharmaceutical ingredients and delivery methods. James, the floor is yours.
Wonderful. Thank you very much, Ryan, and thank you to Life Sciences BC for giving us this opportunity to be able to present Eupraxia to you. We will be going through this fairly quickly, but a more fulsome deck is available on our website. And of course, we always look forward to people reaching out and having interactions. So to give you a little bit of a sense of what it is we do at Eupraxia Pharmaceuticals, we have created a novel drug delivery technology called the Diffusifere. In my background in previous life, as Ryan mentioned, I was a cardiac anesthesiologist and lived my life in transplant. That is all about the precise delivery of drugs. You can have drugs that get to a target, but they need to get there properly. That means you need to be able to get the right dose of drug to the right place for the right amount of time. As you can see in this image here, what we've created is, instead of a lot of the delivery technologies that you see today, which in fact rely on degradation and putting together excipients with drugs in order to be able to have essentially them break apart and slowly release over time. That, however, still results in first-order kinetics, which means basically peaks and troughs. The peaks are where you get all the side effects and you lose the efficacy in the trough. What we've designed here, if you think of them as little balloons about the size of a grain of sand, protected by an extremely strong group of patent families with composition of matter out deep into the 2040s. What this does is allow us to deliver drugs exactly where we put them for a long period of time in a very flat and stable fashion. So what does that look like? What you're looking at here is actually human data from our 300-patient first pivotal trial in osteoarthritis. We'll talk more about that later. What's important here to look at is the stability of the drug delivery. This is from a single injection at day 0 into the patient. You can see there's a very short period of a week where there's equilibration between the inside of the microsphere and the outside where we want the drug to be. But you can't really overdose because you're just getting a concentration gradient flat. Because of that, we're able to deliver over a very long period of time. Here you can see for 6 months with very stable pharmacokinetics. That allows us to have very favorable safety combined with favorable and durable efficacy. So let's talk about this in a couple of different applications and throw some data. So first, let's talk about eosinophilic esophagitis. If you haven't heard of that before, that's okay. It's actually a fairly recent disease. When I went through medical school, we did not talk about EOE because it didn't exist. Instead, these patients were people who were suffering a lot with difficulty swallowing, food boluses, meaning food actually gets stuck right in there. People are often vomiting, and they have a lot of pain with swallowing. Since the diagnosis of the disease, there's been a rush to try and figure out how to do the treatment. Off-label, people get oral steroids, which work quite well, but they're very difficult to deliver with lots of side effects. On the other end, Sanofi has a drug called Dupixent. It's $100,000 a year, has a fair number of side effects. A patient needs to inject themselves once a week and is not demonstrating the efficacy that physicians and patients are really looking for. And so what we've got here is using our technology, using the active pharmaceutical ingredient, fluticasone propionate, is actually injecting directly into the esophagus. So gastroenterologists, when they follow these patients, they see them once a year. and they go down and they do an endoscope and they take biopsies. What we're saying is to put into that treatment algorithm, or that sort of monitoring algorithm, a treatment, so that patients only require treatment dosing once a year, given by the physician while they're already there. So in order to explore the dosing that we would need to be able to achieve that, the top key opinion leaders in the world in this space, led by Dr. Evan Dellon, essentially what you see here is we're dose escalating. The first 3 cohorts, which we've only just reported out, those first 3 cohorts are what we would call the low dose cohorts. And so they are looking at the endpoints, both of how patients feel, as well as the biologic endpoints of what's happening to the disease. What you can see here is that even at these very low doses, where we did not expect to see efficacy really happening, but thought it was more about delivery, pharmacokinetics, and safety, we are seeing not just exceptional safety, but also what we are seeing is in patient-reported outcomes that you can see patients are feeling much better. So of the 9 patients treated to date, 8 of them are feeling incredibly sort of resolved in terms of their symptoms. At the same time, as we are seeing, as we are dose escalating through this, the biological improvement, both in terms of eosinophil counts and histology scores. So again, much more data is available on our website, and happy to talk about it more. This has been a very, very interesting program with lots more data that's going to be reading out throughout 2024. Can also pivot into the other area where here we're talking about osteoarthritis. Here is a single injection into the knee, so an intra-articular injection for the treatment of pain and dysfunction of osteoarthritis. This is our first pivotal trial that we did. It is 300 patients. It is a single dose versus placebo, where we looked at pain and dysfunction, so both stiffness and lack of movement, looking at the WOMAC scale. What's really, really important here is a combination of efficacy, duration, and safety. When patients get osteoarthritis, they tend to have pain daily for the rest of their lives until they get a joint replacement, which you want to put off as long as you can. So what did we see in this trial? I showed you the pharmacokinetics, which where we got a very flat, stable drug delivery for a long period of time. And that resulted in what you would expect, which is a very meaningful efficacy. So what you see on the left is a total WOMAC score. So that means that its stiffness, its function, and its pain with statistical significance to 20 weeks. At the same time, in terms of pain, you are seeing statistical significance to 17 weeks and a clinically meaningful drop of still 2.5 points all the way out to 24 weeks. Now, 70% of the patients who have osteoarthritis have moderate disease and need to be treated for 5+ years before they're eligible for a joint replacement. And so it's really important, and we pre-specified actually looking at the moderate patients. What we see here is not just looking at those moderate patients, but asking the question the Osteoarthritis Research Society International says, which is what is actually clinically meaningful for these patients? Well, a strict responder is defined as a 50% improvement in pain, combined with a minimum 2-point improvement in pain. And we see that with statistical significance over placebo to 22 weeks. So all of this is showing a deeper level of efficacy and a longer duration of efficacy than has really been seen before. But what's critically important here is that there is not today, whether an oral medication or injectable, something that is really safe and efficacious for continuous long-term therapy. Well, we demonstrated in diabetic patients to be no different than placebo. We do not see systemic complications, nor are we seeing cartilage damage. And that's incredibly important because what that means is this has the ability to be something that whether it is lasting 6 months, 4 months, or 9 months, it's the fact that whenever a patient is in pain, whether they're a diabetic, whether they have disease in more than one joint, that we would have the ability to be able to provide relief with an injection at any time the patient needs it and to do so safely. We had a very successful end of phase 2 meeting with the FDA. We have a single phase 3 to do before this drug is approved, 700 patients. It's essentially a rinse and repeat of the data that you just saw. So the clinical risk in this program has really been dealt with the first pivotal trial. We're doing the same thing, but with more power. And that power is really just to fulfill the safety database. In terms of the safety, there's great agreement with the FDA that we have demonstrated local and systemic safety. And if we continue to do that, that this will really be a safe and effective drug for continuous therapy, providing patients with relief in osteoarthritis for many years. We believe that'll be transformational for patients and open up an entirely new commercial market. Overall, we have a lot of data to deliver in 24, lots more in 25. We have a recent uplist to the NASDAQ with EPRX being our symbol. We have $50 million in treasury at the present day and look to a fairly long runway to be able to deliver this data with the cash that we have on hand, and also to be able to use our technology to bring new products to the forefront as well. So with that, thank you very much for giving me the time and the opportunity today, and look forward to speaking to anybody who has interest in this. Thank you.
Thank you, James, for that wonderful presentation. And just also a reminder, it's been in the chat, that many of the companies presenting today are available on the BioPartnering app. So please look them up and send them a meeting request, or you can contact them through the information in these booklet. It is now my pleasure to welcome Dr. Jay Kulkarni, CSO of Nanovation Therapeutics. Nanovation Therapeutics is a one-stop shop solution for lipo nanoparticle and nucleic acid solutions that overcome the fundamental limitations of current technology. Nanovation focuses on extrahepatic delivery systems that are designed in a safe and effective manner to transform the therapeutic concepts into clinical reality. Over to you, Jay.
Thanks, Ryan, for the introduction, and Claire as well for helping organize and sneaking me into the system a little bit late today. So it's my pleasure to introduce Nanovation Therapeutics. My name is Jay Kulkarni. I'm the CSO and co-founder at Nanovation. Before I get into the details about our technology, I just wanted to provide a brief intro to who we are. We're a platform company headquartered in Vancouver, BC. Chairman of our board is Peter Cullis, generally referred to as the founding father of LNP technology. Dom Witzigmann, our CEO. Early on in our existence, we also hired in Wendy Lamson as their chief IP officer, and you'll understand the specifics of that in just a moment. Don Enns is our chief business officer, who is formerly the CEO at Northern Lipids, and then VP head of global drug delivery at Evonik. We also engaged with Molly Stevens in the UK, now at Oxford, as a key opinion leader in the nano space. And one of our key founding members also is Dr. Marco Cipollini, who was quite instrumental in the design of molecules such as KC2, and that eventually led to structures such as MC3, which are lipids used in commercial formulations. And you know, our goal really is to bring together the right set of expertise to help generate the next generation of nucleic acid therapeutics, improving on technologies that currently exist. And just to give you a flavor of what that could look like, a quote from Peter in Nature Reviews Materials that says, almost any disease you care to name can potentially be treated using an RNA-based gene therapy. And so, you know, there's an immense potential in RNA-based gene therapy, but of course there's critical limitations in the delivery and how that's enacted. In the image that I show on the left, the first approved, first ever RNA interference product called Onpattro was a lipid nanoparticle that encapsulated siRNA and delivered to hepatocytes in the liver following intravenous administration. It largely formed the basis for current LNP technology. And you know, what we've seen with the mRNA vaccines, for example, transition of to newer lipids that really help to boost the potency and really, I mean, incredible achievements with modifying the system to provide durable and robust immune response. However, both of these systems, when they're intravenously administered, still accumulate in the liver. And so one of the key pieces to address was, if you wanted to get outside of the liver, you had to re-engineer the system to be able to get to those, get to other tissues as well. And so what we've focused on within Nanovation is to try and address this particular need. In addition to a more, let's say, business or let's say a technological limitation, and in particular, the current approach that's typically used in order to commercialize an LNP or a lipid nanoparticle RNA formulation is that developers have to access technology from a number of different suppliers, and this can add up relatively quickly. It's a pretty complex process, easily done, of course, in the context of pandemic, but now that we've moved on from that, the ability to do this in a more effective manner is not as straightforward. And so what we're building within Nanovation is a toolbox that contains a number of these different pieces, and we break the technology down really into four main pillars, or four drawers, if you'd like. The specialty ionizable lipids, these are molecules that are effectively what help to promote encapsulation of nucleic acids and also intracellular delivery. And so designing these with safety and efficacy in mind, so thinking about how they turn over once they've done their job. The second piece is on LNP composition. So when you combine 2 or 3 or 4 different lipids, what are the ratios in which those lipids are combined, and how that can be leveraged in order to enable delivery to extrahepatic non-liver tissues. Surface modifications. So how do you, during the pandemic, the polyethylene glycol lipid component received quite a bad rap, and whether or not that's justified is a different conversation. But generally speaking, these surface modifications were intended to improve the stability of the particle, and in turn have received a sort of negative view. But our perspective on this is, if there's a fourth component that's to be added and needs to perform that function, can it also be leveraged for functional elements rather than stability alone? And so improving both the tolerability and targeting of these delivery systems, leveraging that fourth component. And we do have earlier and more research-based efforts on RNA modifications. The idea here being, how do you manipulate the chemistry of the RNA in order to increase the amount of protein that comes out of each RNA molecule? And all in, we're hoping to build Nanovation into a one-stop shop IP portfolio to support commercialization of our partners. And almost everything that we do, that we look to partner out on, we have a very strong focus on the patentability of the technology and the ability to commercialize. Freedom to operate is a critical consideration. Just to give you a quick flavor of the types of technologies that we have within our portfolio. The first one that we call the LT-LNP, which is the long circulating LNP, is really the basis for how we access extrahepatic tissues. The traditional on-patch composition or 50-10 composition, when imaged under cryo-electron microscopy, looks like this colored in circle. We've developed a new formulation that has extended circulation profiles and displays a dramatically different morphology, where you see the sort of clear bilayer structure and a overall different morphology. When intravenously administered, these formulations also display different biodistribution and clearance profiles. So when we look at the image in the middle here of a SPECT-CT demonstrating where the particles end up, we see with the typical LNP, you hit the liver quite clearly. With the LC-LNP, though, there's dramatically less that accumulates in the liver. The spleen lights up at this particular time point, the heart lights up, but that's not because of true delivery to the heart. It's that there's still a considerable amount left in circulation. And when assessed using, a radioactive label that doesn't exchange from the particle, what we demonstrate is that circulation time goes from, instead of 15 to 30 minutes, out to 2 to 4 hours. And we've now even developed formulations that go out to 8 hours. And this extended circulation is really what enables delivery to extrahepatic tissues. And we've focused on not using things like cationic lipids, anionic lipids, molecules that are generally considered potentially toxic or challenging to work with downstream, but rather just focused on really modifying the ratios of the components so you can really make this LC-LNP using the exact same 4 components that are in a clinically approved formulation. And that composition is really what drives the distribution and clearance. The impact of working on these sorts of formulations is demonstrated here. So on this slide, I show the impact of leveraging both our LC-LNP technology combined with our own lipids. First, the LC-LNP compared to a benchmark. When we deliver luciferase mRNA and we look at the activity in the bone marrow, we see a dramatically improved activity with the LC-LNP compared to the traditional system. But then once we start to mix and match that with our own ionizable lipid library, we've started to see, up to a 5-fold improvement in activity in bone marrow. And so this sort of mix and match approach is what we've leveraged to really design next-gen therapeutics. And I just want to clarify on this point that as a result of this mix and match approach, it's not that we get 100% of the delivered dose to the bone marrow or anything like that, but we do see improved delivery to the bone marrow. We see improved activity in the bone marrow. We do see some residual activity in other tissues, but manipulating the cargo in that context has worked quite well to improve tissue specificity. And so in terms of how we've partnered and worked with different groups, we're a platform company and we're not in the position to vertically integrate. We're not looking to do that. Our goal is to really focus on our sweet spot, which is this early stage prototype development where we bring the synthetic chemistry expertise and mRNA and formulation together, assess the activities in early preclinical models, and then work together with partners to drive that forward. And so our 3 main goals for BioUS this year are looking to meet with potential partners and biotech pharma interested in developing these nucleic acid therapeutics. We're looking to meet with CDMOs for downstream scale-up and manufacturing considerations. Our focus here really is on fully integrated CDMOs. And then finally, meeting with regulatory groups that can help us direct our regulatory affairs efforts. So with that, I'd like to thank you all for listening and the opportunity to present today. Thanks, Ryan.
Perfect. Thank you, Jay. That was an excellent presentation. And to finish off the presentations for today, we have Lillian Zhang, Senior Associate Business Development, Acuitas Therapeutics. Vancouver-based Acuitas Therapeutics partners with pharmaceutical and biotechnology companies, as well as non-governmental organizations and academic institutes to advance nucleic acid therapeutics into clinical development and commercialization. Acuitas's clinically validated lipid nanoparticle technology enables Alien Pharmaceuticals Onpattro and Pfizer-BioNTech's Comirnaty, and their current focus includes genome editing, epigenetic medicines, therapeutic antibodies, and vaccines, including improved multivalent and novel therapeutic vaccines. Welcome, Lillian.
Thanks, Ryan, for the introduction, and thank you, LSBC, for having me. It's my pleasure to speak a little bit more on Acuitas, provide some background on the company, speak on our technology, and also our goals and focus for bio.
Sorry, Lillian, just let you know your presentation isn't in full screen mode.
It is not in full screen mode. Okay, I was just going to say thank you. Let me try it this way. Is that working?
That's perfect. Great.
Okay,
thank you.
fantastic. Thank you. All right, so sort of diving right in here and providing a little bit of background on the company. At Acuitas, we're purely a lipid nanoparticle developer and licensor. And so that means we don't do any sort of internal product development in competition with any of the parties that we do license our LNP delivery technology to. And so we work with a variety of organizations, from big pharma to small biotechs to academics and to foundations. And we're able to support our collaborators all the way from their discovery work to commercialization. And we currently have 2 products on the market using our LNP technology, and that is Onpattro and Comirnaty. So providing a little bit of background as to why LNPs, the core payload that we work with is nucleic acids. And nucleic acids, when injected into the body, they are very fragile, so they can break down very easily. And they're also very large molecules, so they're not able to get into our cells. And so with the LNPs, they're able to package the nucleic acid and protect it as it goes through the body to our cells. And because the LNPs are very small molecules, they are able to get into our cells, release the nucleic acid in the cytoplasm, and allow the nucleic acid to go where it needs to go, typically the nuclease in the cell, to perform its function. So a very simple analogy is, you buy a very fragile glass ornament representing the nucleic acid, and the delivery service would be our LNP. It would package the glass ornament and keep it safe no matter how rough the ride. And it would bring it to your door, but not only leave it on your porch, but let itself into your home and place it exactly where you would want it on, for example, your Christmas tree. And so I want to touch a little bit on the technical capabilities we do have at the company. So all 6 of these areas that you see here are internal to Acuitas, and they do allow us to support our collaborators from end-to-end drug development. And so starting with our chemistry team, our LNP improvements are due to improve lipid composition. So we do have a very strong chemistry team that looks at design and synthesis of novel lipids, mainly looking at cationic lipids, but also PEG lipids. And then our strong formulation team, they take these components from our chemistry team and they look at LNP formulation and characterization and creating our LNPs. Our formulation group is also responsible for formulating all of our collaborators' payloads in the preclinical phase for all of their preclinical testing. Our analytical group, as you can see, they do bioanalytical method development and transfer. So that means testing the LNP itself, as well as the LNP with the nucleic acid, and looking at all of the various characteristics to make sure that everything is looking as it should. Our preclinical group, they're responsible for all the pharmacodynamic and safety screenings, which we all do in vivo, as well as looking at PK/ADME. They also run our core NHP research program that we have here at Acuitas. So screening all of our LNPs through mice, but also through NHPs to ensure we have the relevant data to provide to our collaborators when the time is right. Our CMC team is very key because we don't do cGMP manufacturing here at Acuitas. And so when our collaborators are ready to take our LNPs into the clinic, our CMC team would conduct the technology transfer of the chosen LNPs to either CMOs in our network, CMOs that they would like to work with, or even sometimes to our collaborators' facilities. And they would ensure that the LNP continues to meet our quality standards, that the CMO or the collaborator is set up with the right equipment, processes, and so forth. And lastly, regulatory. A lot of our senior members at our company, they have a lot of experience in IND and NDA submissions. And so this has really allowed us to provide regulatory advice and assistance to our collaborators as well, especially in the LNP space. So I want to touch a little bit on our approach to continuously identify and improve and refine our LNPs. The process is illustrated quite simply in row 1 here. You can see it's sort of a 4-step process. So we start with lipid design and synthesis of new ionizable lipids by our chemistry team. And then these lipids are formulated into LNP by our formulation team, who then determine the biophysical characterization of the formulations. And then this is further looked at by our preclinical group, allowing us to look at the potency and the safety and comparing that with the biophysical characterization and also the development structure to develop that SAR. And so we're continuously repeating this design testing process that you see here, using this refined SAR model to continue to predict additional active structures. And so some of the fruits of our labor are shown in row 2 there. And that graph is showcasing the in vivo expression of luciferase in the liver of mice. And so the luciferase expression is on the y-axis, and all of the LNPs are on the x-axis. And I shouldn't say all of our LNPs because we have over 1,000 LNPs in our library. So it would be quite overwhelming to graph all of them. So we just graphed a select few to showcase the performance. And I would like to specifically call out 2 of our publicly known LNPs, MC3, the little green bar on the very left-hand side there, that is used in Onpattro, and 315, the black bar right in the middle of the graph that's used in Comirnaty. And so we do like to compare our LNPs against MC3, which is a great LNP that was developed back in 2009. But since then, we've been able to significantly improve our LNPs. And now they're about 15 to 20-fold higher in potency and therapeutic index. And so, as you can imagine, most of our current collaborators are using our newer LNPs on the right-hand side. So these are the 5 areas that we mainly work in with our collaborators. They involve gene editing, vaccines, protein replacement, antibodies, and epigenetic medicine. And so we're always very happy to take on new collaborators in these 5 areas or explore new areas of interest with collaborators as well. And just quickly showing some of our first in-partner successes. So as you can see, we have first RNA interference-based medicine, first mRNA-based COVID-19 vaccine, and a number of first mRNA genome epigenome editing. And so Acuitas has been quite fortunate to work with a number of great companies, and we hope that this success continues to live on. So we do regularly publish in a number of leading scientific journals. You can visit our full publication website, full publication list on our website at acuitastx.com. I unfortunately don't have the time today to go into any publication in specific detail, but I do want to point you to that last category there, the non-hepatic applications. So as you have already heard, it is a very hot field in the delivery space right now. And so we do have a number of publications on targeted LNP delivery that I would invite you to check out. So Acuitas has also received global recognition in terms of our founders and our company. And so a number of our founders have received the Governor General's Innovation Award. The company has received LSBC's Company of the Year Award, the Greater Vancouver Board of Trade Revenue Over $5 Million Award. And our president, CEO, and co-founder has received Biotech Canada's award, as well as EY Entrepreneur of the Year Award in the Pacific West. And one of our co-founders, Dr. Peter Cullis, has received the Gardner Award, the Tang Prize, and also the Bloom-Burton Award. So to wrap things up and illustrate why we believe Acuitas continues to be the leader in the LNP space, and that is because we do believe we have first-in-class and best-in-class LNP technology with a very broad, comprehensive portfolio of IP, including hundreds and hundreds of patents. We have a number of cGMP-ready lipids pre-run at a number of CMOs and have a lot of expertise in this tech transfer process. As illustrated, we regularly publish in a number of top journals. And just a little statistic to end off, we've advanced about 26 drugs into the clinic in the last 2 years. And so zooming specifically in on bio, we will have representation from 2 departments at Acuitas at bio, and that includes our business development department, who is on the partnering app, as well as our operations department. And so we are looking mainly at 3 main objectives for bio this year. The first one being to seek out new opportunities to out-license our LNP delivery technology, as well as new collaborators to enable further nucleic acid product development. And we're also looking to check in on our existing partnerships to ensure that the relationship remains healthy and successful. And last but not least, we're looking to keep up to date on the industry knowledge through the conference itself. And our operations team is looking to connect with suppliers and vendors to evaluate opportunities for operational improvement. And lastly, we are also looking to help raise the profile of the BC Canada life sciences ecosystem. And so this is my last slide. You can reach out to me at bd@acuitistx.com if you would like to learn more about Acuitis or if you would like to connect. And our website is also listed there, acuitistx.com, if you do want to learn a little bit more on our technology and view our list of publications. Thank you so much, everybody. Back to you, Ryan.
Great. Thank you very much, Lillian, for that great presentation. And that's wonderful to see. Got some great goals for bio. Also wanted to thank our other speakers today, Dr. Emily Chen, Dr. Anthony Feja, Suzanne Gill, Dr. Hal Gunn, Dr. James Halliwell, Dr. Jay Kulkarni, and Deputy Minister Fazil Millar. Also, thank you as well to our Bio San Diego supporters, whose support is invaluable, including our BC Bio San Diego supporter, the Government of British Columbia, the BC Networking Reception event sponsors, Acuitis Therapeutics, and Invest Vancouver. And as I hope that you have learned from today's presentation, BC is home to some very exciting leading innovations with the potential to advance patient care and outcomes. All the companies who presented today will be at Bio in San Diego, in addition to 40 other BC-based companies. To learn more about our BC delegation, please note that the QR code on the screen. A link to this information was also shared in the chat and will be sent to everyone in a post-event email tomorrow along with the recording. If you are planning on attending Bio, please feel free to reach out to ourselves, Life Sciences BC, and any of the companies and organizations that presented today or were part of this event. Where our contact is on the Bio Partnering app, as well as in the booklet that has been today. So thank you again for attending Life Sciences BC's BC Delegation Spotlight for Bio San Diego, and I look forward to seeing everyone in California in just over a week. Thanks very much, and take care.